Muna, we are set out to identify novel therapies that are targeting neuroinflammatory processes in order to develop therapeutic effect against neurodegenerative disorders. We are doing this by combining two platforms with each other. First of all, we're building on a target discovery, target validation platform that is really an all-in-human platform. We're utilizing here the genetic information that I referred to earlier, pointing towards specific genes and gene factors. What we are building further on this by also looking at differential gene expression. We're doing that by spatial transcriptomics that we're pursuing in human-derived brain material from both Alzheimer's disease patients. But we're comparing this with brain samples from subjects that actually do show resilience towards amyloid pathology. Important to note here is that this is seen in a significant amount of postmortem brain samples where, despite the appearance of amyloid pathology comparable to what we see in Alzheimer's disease, subjects remain cognitively normal, hence the designation as resilient. And what we're particularly interested here is in differential gene expression with regard to proximity to pathology to identify genes that might be correlated either to cellular vulnerability around pathology, but also towards neuroprotection resilience around or in relation to neuropathology. We are then integrating these findings with other omics databases, especially the genetics databases I referred to, but also protein-protein interaction databases, in order then to funnel down the amount of potential targets to a educated guess for the most relevant and the most attractive novel drug targets that we're then taking through a validation platform, starting in vitro, again working with human cell lines, in order to then to progress this into an in vivo context where we utilize the same human stem cell derived cell lines that we are grafting into the brain of mouse models that carry certain features of human pathology, again, like, for example, amyloid-beta pathology, but this can also be taken forward into other contexts in other neurodegenerative disorders. That then allows us to study human-derived cells in the context of a human pathology in an in vivo model. So that's the target discovery, target validation, all in human platform. And then in parallel, we're pursuing a structure-based drug design approach where we are building structural biology information around the particular targets that we identify. This has been done by crystallography as well as cryo-EM studies that then allows us to use the structural information in order to build in silico design of selective and potent compounds and then validate these in experimental settings in order then to further mature the modeling further as well as design of the compounds further. So it's these two platforms that we bring together to target neuroinflammation.